Hello friends, welcome to SR Academy.
Welcome to SR Academy. Empowering minds, igniting curiosity. Your gateway to knowledge unleashed. Let's get started.
Today we are going to discuss tissue engineering scaffold in this video. So tissue engineering scaffold. So scaffold we have discussed in module 1. We came to know that the scaffolds are nothing but a synthetic materials or a synthetic, or you can say artificial ECM. So the scaffolds are usually used to do the function of what ECM does. So what is ECM? So extracellular matrix, so which is present in our tissues, right? What is it doing? What are the functions of the ECM? It gives the structural and mechanical support, right? So the same function the scaffolds has to do. So and why do we need scaffolds? Because if there is any wound, or if there is an because of accident, if a lot of tissue has been damaged, then we need to rebuild that particular tissue. So to get the extra tissue, you need to have extracellular matrix. But as of now, naturally, since the all whole cells has been destroyed in that particular damaged area, we won't be having ECM. So we can we can develop the scaffold. So scaffold will act as a ECM, extracellular matrix, which can be synthesized synthetically, okay, with the help of engineering principles and methods. So now with this scaffold, so what we have to do, we have to seed the cells. That means whatever the cells we need to develop, whatever the tissue we need to develop in that particular damaged area, those cells we need to take and we need to implant or seed inside the extracellular matrix. Along with the cells, we will be adding growth factors, so that it will facilitate the cell division and cell proliferation and cell division, and finally growth. So once the cell is seated inside the scaffold or artificial extracellular matrix, so what happens? The cell will attach on the scaffold. So once the cells are attached on the scaffold, the cells will start dividing. So where the growth factors also will, or biological active compounds or factors, they will increase the speed of or a rate of division, okay, so that the cell differentiation will be faster and cell will start growing, and finally it will give you the tissue. Right now that tissue has to have some particular structure. So that structure is decided by this particular scaffold or artificial extracellular matrix. Now this particular tissue which is generated, now I can implant back into the damaged part, damaged liver or damaged kidney, or even damaged normal muscles, so that the particular part which is missing can be regained by this new tissue. And finally, when the new tissue is formed, now the scaffold has to undergo degradation, so that the tissue will develop its own extracellular matrix naturally, and whatever the synthetically we have implanted a scaffold, that has to degrade over a period of time. So that's how we need to develop the scaffold. So that is what we will be studying in this particular topic. So that's it. This is the general brief introduction. Then we'll be talking about different ideal characteristics of scaffold and what are the requirements, all other things we will study. Okay, so let's start. Let's begin the topics. The scaffolds mimic the extracellular matrix of the native tissue. That means whatever it copies the extracellular matrix function, right? So scaffolds are materials that have been engineered to cause a desirable cellular interactions to contribute to the formation of new functional tissues for the medical purposes. Understanding? So scaffolds are materials that have been engineered, right? That have been engineered in the sense they have been synthesized or by using engineering principles and techniques. For what purpose? To cause desirable cellular interactions. So that means so that the cell can come and attach to on them, and then they can divide. Okay, these are all called as desirable cellular interaction. To contribute, for what purpose? To contribute the formation of new functional tissue. So to contribute or to make the new tissue for medical purposes. Understanding? So that is how we can define scaffolds. So cells are often seeded into these structures, that means scaffolds capable of supporting three-dimensional tissue formation, so that you will get the three-dimensional structure of the tissue. Understanding? So the scaffold gives a structural support, mechanical support. Here we are talking about structural support, three-dimensional, how exactly that particular organ should look like or a tissue should look like, that is also decided by the scaffold, right? Three-dimensional tissue formation will be there. Okay. So key functions of scaffolds, they usually serve at least one of the following purposes. What are those purposes? There are 4 purposes. Any one of the purposes is enough depending on what the particular patient requirement is there. So first thing, it allows cell attachment and migration. That means the cells, whatever the seeded cells are there inside the scaffold or extracellular matrix, so it has to allow them to attach on their cell surface, and the migration. Okay, so migration in the sense, as the cell divide, cell will start migrating, no, like that. So next, either that was about the first thing. Next, deliver and retain cells and biochemical factors. That means you need to deliver and retain the cells in that particular, it should hold the cells, and biochemical factors. Biochemical factors are nothing but, we are having bioactive factors. Bioactive factors are nothing but growth factors. Understanding? So it should enable diffusion of vital cell nutrients and expressed products. That means it should have capability to diffuse a vital cell nutrient. That means whatever the nutrients cell requires for its growth, it should allow them to diffuse through their structures. Okay, because cell has to divide, right? For that reason we need certain nutrients, so those nutrients should be allowed. So it should exert certain mechanical and biological influences to modify the behavior of the cell phase. That means it should have some mechanical and biological properties to modify the behavior of the cell phase. Now the history. So in 2009, an interdisciplinary team led by the thoracic surgeon, that is Thorsten Wallace, implanted the first bioartificial transplant that provides an innate vascular network for post-transplant graft supply successfully into the patient awaiting tracheal reconstruction. For tracheal reconstruction, whatever the vascular network was needed, that was designed by the artificial scaffold, whatever we discussed so far, with the help of those things. Okay, so in 2009, that was the first successful bioartificial transplant. Okay. Requirements. What are the requirements for this particular scaffold? I mean, if I need scaffold to be used in tissue engineering, what it should meet some requirements. The first requirement is biocompatible. Obviously, any material you are inserting inside our body should be biocompatible. That means only specific function it has to do. It should not cause any toxicity to any other parts of the tissues or the cells. The next thing is high porosity and adequate pore size. So what are the high porosity means? It should have a lot of pores inside it. Why? Because it should provide vital nutrients to the cells for their growth, and also it should have it should allow the particular growth factors or biological active compounds or factors or chemically active compounds should be there. Okay, so for the diffusion of specific cells and the nutrients. Next, should provide structural integrity until new tissue is formed. That means it should provide three-dimensional structure until the new tissue is formed. So next, the fourth one is biodegradable. That means it should avoid surgery to remove, so that we need to, this particular scaffold should degrade as the new tissue is formed, or as the new structure is formed, the tissue has to degrade itself. So these are the 4 requirements: biocompatibility, high porosity and adequate pore size, structural integrity, biodegradability. So next, materials and properties. What are the materials and properties we are talking about here? It depends on what kind of materials you are using to develop this scaffold. So materials needed for each application is different, definitely. So dependent on the desired mechanical properties of the material, we are having a different scaffolds. For example, tissue engineering of bone, for example, will require much more rigid scaffold. That means a little bit hard or a tough scaffold compared to the tissue. We have scaffold which is used for skin generation, which requires very smooth one, right? So there are few versatile synthetic materials are there which we will be using as scaffold. We are having PLA, PGA and PCL. So they are used for many different scaffold applications. So synthetic materials, one of these commonly used materials is a PLA, that is polylactic acid, a synthetic polymer. and we are having this PLA, that is polylactic acid. This is a polyester which degrades within a human body to form lactic acid, and naturally occurring chemical which is easily removed from the body, right? Understanding, once it degrades, it forms the lactic acid that will be removed easily from the body. So similarly, materials are there, similar materials are there, for example PGA and PCl. So PLGA, so it's one of the this thing material which is used for the scaffold. This PLA is commonly combined with the PGA. That means PLA and PGA are both combined to create PLGA, that is polylactic glycolic acid. So this is specifically useful because of the degradation of the PLGA can be tailored. That means we can monitor. So the degradation rates and all, if I need a lot of time for degradation, I can increase the time by varying the composition, okay, by altering the weight percentage of the PLGA, PLA and PGA. For example, more PLA, slower the degradation. For example, for wound healing, we need slower degradation because it takes a lot of time. So slower degradation, and for faster degradation I can go for example, for skin related issues I can go for faster degradation, so that the skin, since the cells are very smooth, they can form faster. When you are talking about bone and something like that, it requires a lot of time to heal, so I need slower degradation. Understanding? So whatever the degradation rate I need, slower or faster, I can manipulate, I can tune, or I can tailor. Okay, all are same, all words are meanings are same here, so that I can have whatever I need. If I need a slower, I can have that. If I need faster, I can have that. So I can basically degradation rates can be manipulated. So this tunability, so along with its biocompatibility, makes it extremely useful material for the scaffold creation. So PLGA plays a very important role because of its tailoring properties. So natural materials, that was about the synthetic materials and example. Natural materials, scaffolds may also be constructed by using natural materials. In particular, different derivatives of extracellular matrix have been studied to evaluate their ability to support cell growth. That means we are having a ECM, different derivatives are there or components are there in the ECM. With the help of them, we can create the scaffold. So that will be our natural scaffold. So we know that we already studied what is ECM and what are the components, soluble and insoluble components we have studied, right, in previous module. So protein based materials such as we are having the collagen or a fibrin or polysaccharide materials like chitosan, or even we are having GAG, that is glycosamine glycans, that we studied in detail in the previous module. So they all have proved suitable in terms of cell compatibility. That means the protein based materials, these are some of the examples, these all are what? ECM derived compounds. So these all can be used for what? As a scaffold material. Synthesis of scaffolds. How we can synthesize the scaffold? There are plenty of methods are there to prepare a porous structure to be employed as tissue engineering structure or a scaffold. That means we can have a different techniques. Okay, all can be used to synthesize scaffold. Okay, every method has got their own advantages and disadvantages. I am not going to explain every material, every synthesis the techniques here. I'll be explaining only two, and that should be sufficient. So nanofiber self-assembly is there, laser assisted bioprinting is there, electrospinning is there, gas forming is there, emulsification, freeze drying is there, solvent casting and particulate leaching is there, thermally induced phase separation is there, CAD and CAM technologies can be used, and then self-assembled recombinant spider silk nanomembranes also can be used to synthesize scaffold. These are the most widely used techniques to synthesize scaffolds based on our requirements. The first in the whole these things I will be discussing about two things, that is first two things, nanofiber self-assembly and laser assisted bioprinting. So in nanofiber self-assembly, what happens? The molecular self-assembly takes place. What is molecular self-assembly? What do you mean by self-assembly? Self-assembly in the sense from atoms to the clusters, molecules to the clusters to the particles, or to the nanoparticles or nanofibers. So that is called as self-assembly. It has to form within itself. There has to be no chemical reaction or nothing like that. So whatever once we add two things, both has to come together and form self-assembly. So molecular self-assembly is one of the few methods for creating biomaterials with the properties similar to the scale and chemistry to that of the natural in vivo ECM. That means it will have the properties which is similar to the ECM. Okay, natural ECM, a crucial step towards tissue engineering of complex tissue. In order to get the natural in vivo extracellular matrix properties, we can use nanofiber self-assembly so that we will get exactly the replica of ECM. Why? Because what is ECM? ECM is again an extracellular matrix that is also one of the structure, one of the biomaterial, right? So this material is also again made up of what? Cells, right? The basic building block of any tissue, right? So those cells can be cloned or used for a self-assembly, so that the self-assembled ECM we will get with the help of nanofiber self-assembly. So that why it has to be fiber? Because ECM is a network, okay, so that it should give the structural support, like it should be like this, three-dimensional structure support. So that's how it is nanofiber self-assembly. Named as nanofiber self-assembly. So moreover, the hydrogel scaffolds have shown superiority in vivo toxicology and biocompatibility compared to the traditional macro scaffolds and animal derived materials. That means when compared to the other techniques, the hydrogel scaffolds with the help of nanofiber self-assembly technology which has been produced, they showed a superiority in vivo toxicology and also biocompatibility compared to the traditional macro scaffolds and animal derived materials. Whatever the conventionally it has been used for this particular as a scaffold technologies or the techniques, so that when you compare that with the nanofiber assembly, so here one of the example, that is hydrogel scaffold, is showing the superiority in biocompatibility and toxicology studies. So next topic we are having the next technique we are having laser assisted bioprinting. In 2002 there was some study which was conducted by Koch et al. They focused on whether the laser assisted bioprinting, that is lab BP, can be used to build a multicellular 3D patterns in a natural matrix, which means 3D printing, okay, and whether the generated constructs are functioning or forming tissue. So that was studied in 2012, that is very recently. And LABP, this is nothing but what laser assisted bioprinting arranges small volumes of living cell suspension in set of high resolution patterns. So bioprinting, all of you know, 3D printing and 3D bioprinting, there is a difference. So in biology for engineering study, I have explained it in detail in biology for engineering playlist. Video is there. You can just go through that video so that you can understand what is the difference between 3D printing and bio 3D printing. So what happens in bio 3D printing? We will be printing in normal printing, we will be using ink to print, whereas in bioprinting what happens, we will be using cells instead of ink. That's it. So LabVP arranges small volumes of living cells suspensions in set high resolution patterns. That means where the ink was used in normal 3D printing, here cell suspension will be used. What are the cells need to be developed in the scaffold? Understanding. Now the investigation was successful. The researchers foresee that the generated tissue constructs might be used for in vivo testing by implanting them into animal models. That might be in the research. So that was about the the scaffold and their history, their structure, their key components, and how it can be synthesized. Okay, tissue engineering scaffolds. So if you have any doubt, please you can write it to me. The next topic we are having types of biomaterials that we will discuss in the next class.